What's going on, everyone? Welcome back to another episode of the Muscle Memoirs Podcast. We have a terrific episode for you today. Join with me across the screen is Dr. Sarah Berry. Dr. Berry, how are you?
I'm good. Thank you for having me, Mike.
Absolutely. Before we get into our topics of discussion today, for the listeners who might not already be familiar with you and your work, could you give us a little bit of background about yourself and perhaps what you're currently working on?
Absolutely. So I'm a nutritional scientist at King's College London. I'm also the lead nutritional scientist on the Predict program studies, which I know we're going to dive into today. My specialist area of interest over the last 20 years has been all around postprandial metabolism and postprandial lipemia, which again, we can do a bit of a deep dive into, but very much in the context of how diet affects cardiometabolic risk factors. So how dietary components and whole diets affect heart disease, type 2 diabetes, and anything related to cardiometabolic health.
Excellent. So as you alluded to there, basically all of the data that we're going to be discussing today stems from this Predict One study. It was absolutely fantastic. We had on Dr. Anna Valdez in a previous episode to kind of go over the general bits of the study, but it was many episodes back, and we've accumulated a bunch of new listeners since then. So I think it is worth an update for those who might not be familiar. And of course, we'll link the study in the show notes as well for those who want to get into the nuances. But give the audience a nice overview of what the Predict One study was.
Okay, so the Predict study forms part of our Predict program of research, and this is the world's largest personalized nutrition program to unravel how we respond to food, how much variability there is in our responses to food, and what's actually causing this variability. And I often think of it in a nutshell as really pushing the fast forward button on nutritional research and really forging the way ahead in terms of personalized nutrition. And I think before I kind of go into actually what we did with the Predict program of research, if you're happy, perhaps we could kind of sidestep and just talk about why do we even need personalized approaches to nutrition. And, you know, I think this raises an important question that something's obviously not working because we have huge amount of global deaths, huge amount of morbidity that we know are underpinned by dietary related risk factors. And I'm often asked the question, is it because dietary advice isn't working? Is it because we're not following the advice? Now, it's a little bit of both, but also it's because nutritional research is evolving at a huge pace. And it's because we're really starting to understand that we're really complex as individuals. So we have thousands of different biological pathways that are involved in how we process food. We're really starting to understand that food's complicated. So typically in nutritional research, I'm sure you and many of your listeners are aware, we focused on single nutrients, on fat, protein, carbohydrate. But actually food is really complicated. It has about 26,000 chemicals in different foods. And it also exists in this very complex matrix, this very complex structure that modulates its health effects. So if we throw together the complexity of food, the complexity of us as individuals, it only stands to reason that we're all going to respond very differently to the same food. So this kind of one size fits all approach that we've typically taken in the past, you know, may be one of the reasons why, you know, population-based advice isn't working. And I believe is one of the reasons why we really need to start thinking in a more personalized, individualized way. And typically in nutritional research, you know, when we conduct dietary interventions, we typically report the mean response. We typically report on average, this group of people respond in this way and this group of people respond in the other way. But given how variable our responses are to food, we really need to move beyond this kind of one size fits all and looking just at the mean. And I think it's been really hard up till now to do this in nutritional research. So the studies that I've typically run at King's College London have been these very small scale, very precise, randomized control trials, typically maybe in 20 or 50 people. And on the kind of flip side are the kind of big epidemiological studies where you have hundreds or even thousands of people, where you do very imprecise measures, but in large scale in a big population. But actually we're at this really exciting transition, and this is what Predict is really capitalizing on. We're in this exciting transition of where we can collect huge amounts of data, so we can get scale, we can get breadth across a whole breadth of measures, so we can get depth and we can get precision. And this is because we can capitalize in nutritional research on novel technologies. We can capitalize on the use of remote clinical testing, so continuous glucose monitors, on all of these health apps. Most people either have a Fitbit or an Apple Watch or apps on their phone that monitor maybe what they're eating or how many steps they've done. And there's also ability to do a lot of home testing. So this could be sending off stool samples for microbiomes, taking swabs for DNA, etc. And then the exploding area of citizen science. So many people are becoming really aware of how food or lifestyle or health behaviors affect their health outcomes. And so people are becoming more interested in finding out more about how they respond to different foods. And what we've been able to do is bring all of this together with our Predict program of research and capitalize on people's interests, so citizen science, these novel technologies, and this ability to capture huge data. And so with the Predict program of research, it's a very unique collaboration between traditional nutrition academia, so academics like myself at King's College and other academics at Harvard, Stanford, and Tufts University, as well as AI and tech. So there was a company called Zoe who were formed specifically with the purpose of undertaking this program of research. And I think this is what's kind of pushed that fast forward button on nutrition research in the way that I mentioned earlier, because we're combining this very fast-paced, novel AI machine learning approach together with nutrition academia. And so for the Predict-1 study, which is the one that we've been publishing on over the last couple of years, we looked at people's metabolic responses to foods in two different ways. We looked at it in a very tightly controlled clinic setting, but also during a 2-week at-home phase, capitalizing on these novel technologies. And we had 1,100 individuals. We had 100 individuals from the US who were our validation cohort, which is important for machine learning and prediction algorithms. And then we had 1,000 people in the UK. And these were predominantly from the Twins UK cohort, which allows us to unravel and tease apart the genetic contribution to how we respond to food. And these people underwent a whole battery of different tests where during the clinic phase and the at-home phase, we collected stool samples for metagenomic sequencing. We measured postprandial responses using continuous glucose monitors and also dry blood spots for postprandial changes in blood lipids, which we can delve into a bit more detail. We recorded everything they ate and drank for the 2 weeks using our app and a kind of whole team of dietary assessment nutritionists who monitored what people were eating and drinking in real time. And from the blood samples that we collected, we measured a whole vast array of different assays, from metabolomics, where you have hundreds of different measures of metabolites that are circulating. And we put all of this together to look at how much variability there is between people, what's causing the variability in responses to food, and then ultimately build a prediction algorithm to see if we could predict people's responses to food. And I think the headline from all of this research is that we found huge variability. As we alluded to earlier, so we found about a tenfold difference in how different individuals responded to exactly the same food. We found that this wasn't all determined by genetics, that genetics actually only played a really small part, and that how we eat, as well as what we eat and who we are, is really important in determining our responses to food. And I think this was really a really interesting part of this, because we found that actually it was mostly modifiable factors. So we're not a hostage of our genes, that we can actually have control over how we respond to food. And something else that we really focused in on was our microbiome. So these are the trillions of bacteria that exist in our gut. And we've developed this microbiome diet signature as well, where we could see a common set of what we call our good bugs and a common set of what we're calling our bad bugs that were associated differentially with either good or bad dietary patterns and good and bad health outcomes.
Excellent. A finer detail there that I think is awesome and really important for the listeners to understand about the Predict-1 study. And then as we're talking about these individual responses to standardized test meals, these different macronutrient ratios that we're playing with in these scenarios, right? Weren't they all delivered in a muffin? Is that correct?
Yeah, so we fed thousands of muffins over the 2-week at-home phase. I have typically used muffins in all of my research for the last 20 years to look at postprandial responses to food, because it's the easiest way to actually manipulate a nutrient composition within a single food. So we fed a very standardized metabolic challenge muffin in our tightly controlled clinic day, which had 50 grams of fat and 85 grams of carbohydrates. So we could really kind of push the system to look at metabolic flexibility and look at metabolic responses downstream to this high fat, high carb load. And then during the 2-week at home phase, participants were given in random order and in duplicate muffins that differed in the fat, protein, carb composition and fat composition. And they were given these to consume having fasted overnight, and each of these were consumed in duplicate. And this was so that we could look at, as well as individual variability in responses to food, which is the difference between an individual. So the difference between, say, myself and you, Mike, we could also look at what we call intra-individual variability in responses to food, meaning day-to-day differences that I might have to exactly the same food. And this is really important, actually, to be able to really define what causes variabilities in people's responses to food, because for precision nutrition to be effective, we need to understand what is causing the day-to-day variability within an individual versus the variability that occurs between an individual.
Very good. A general question for you here, as you were designing this study and why you felt was it needed, was it more or less a product of the fact that this is an emerging area that's just really interesting, or perhaps maybe there was also a concern for the previous research on it, right? So we did have ZV, right, and that kind of started the discussion on this, and it was really interesting. But also, it seems like Predict One and all of the data we have coming out of this is probably the best available data on precision nutrition. Yet before all of these very recent publications, it seems like all of these precision nutrition companies were already popping up and already making a bunch of very, very interesting claims that perhaps weren't yet validated by the data. Did you see a lot of these things popping up and almost say to yourselves, like, we need more data, and what's currently available isn't sufficient, but we know where this needs to go in the field? Was there any thoughts related to what was going on in that regard?
Yeah, I think that's a really good point of discussion. So the ZV paper, so from Erin Segal's group, was, I think, quite pioneering in terms of, or transformation in terms of how we think about personalized nutrition. And, we were becoming aware that there is this huge variability, so there's huge inter-individual variability. But that was the first piece of research that showed that actually collecting the right scale, breadth, and depth of data, we may be able to predict individual responses to food. Now, what we have known for some time is that there's lots of individual determinants of our responses to food. So there's been lots of study that have been highly valuable in nutritional research over the last 20, 30, 40 years, looking at, for example, how does sleep affect the next day's glycemic response? How does time of day affect glycemic response? How does the meal composition affect your glycemic or your lipemic response? And these have all been really valuable, but what they haven't done is they haven't integrated these together, and they haven't been able to look at the relative importance. And this is what we're doing with the Predict studies. For the first time, we're looking at the integrated and interrelated determinants, because we know that depending on the outcome that we're interested in, different factors are more important in determining that response. So for glycemia, for example, we know that the genetics and the composition of the meal is quite important. But for lipemia, for example, we know that the microbiome is more important than genetics. And this, I think, is the problem with all of these different personalized nutrition companies that have been popping up, or products that have been popping up, is that without having huge data that can look at the integrated responses, they tend to focus on individual components. And in my opinion, as a nutritional scientist, and based on our Predict study data, that's not an appropriate thing to do. So genetic only testing, or microbiome only testing, or CGM only testing, I think that together they're really, really valuable. They give us all the pieces of the puzzle. But I think that as individual pieces of a puzzle, they really lack value. And I think that they're misleading in a way to consumers, because I think that they're making promises that I think are promising more than can actually be delivered. So by just testing DNA, we're seeing a tiny bit of the puzzle. By just testing the microbiome, yeah, we're seeing quite a big piece of the puzzle, but it's not the whole piece. We have to piece together these other bits. And I think that's what's exciting with the Predict program, that I hope that from this data we're already, on a weekly basis, we're fitting one more piece of that puzzle together, which for me as a nutritional scientist is beyond exciting.
Yeah, it really is fascinating, even just following along as you produce new research from this Predict One study. But that all segues really nicely into the first topic that I want to expand on, and leads us into our first recently published article from your group on this trial, and that's looking at dietary inflammation. So before we get into the results of which postprandial metabolic response seems to be more associated with dietary inflammation, can you provide an overview for the listeners of what this is on an acute basis, and why we're concerned about it, and then what can eventually happen in the long term?
Absolutely. I think perhaps it's first important for us to understand what happens when we actually consume food, and to see why this might relate to dietary inflammation. So are you happy for me first to cover a little bit about postprandial lipemia and glycemia to put it into context? Great. So when we consume a meal or consume a food, we typically consume mixed nutrients. So it's very rare, unless you're just eating some jelly beans, for example, you'd ever consume a single nutrient. So we typically consume a meal that contains fat, carbohydrate, protein, and maybe a little bit of fiber in it. What happens is that the carbohydrate elicits what we call postprandial glycemic response, which is a short, sharp rise in glucose circulating in the blood, which peaks around 30 minutes, returns to baseline around 2 hours. The fat in the meal causes a more slower and sustained elevation in circulating triglycerides from the fat in the meal, which peaks around 4 hours, returns to baseline around 8 hours. Now, if we map these excursions in glucose from the carbohydrate in the meal and triglycerides from the fat in the meal onto a typical eating pattern where we consume multiple meals, so 2 to 3 main meals, 2 to 3 snacks a day, we actually spend most of our day in this state of metabolic flux with all of these short, sharp rises in glucose, with this kind of sustained elevation in triglycerides throughout the day. And this is really important because we now know that these elevations in glucose and fluxes in glucose, and that this elevation in triglycerides initiates a whole cascade of downstream events. And one of the most important of these, we believe, is to be this initiation of an inflammatory response. You also have, alongside that activation of oxidative stress, you have changes in hemostatic factors, which is all around kind of how your blood clots. And you also have, from the fat in the meal, changes in circulating lipoproteins. And these are the very specialized particles which carry the fat around our body. So people may have heard of HDL, which is high density lipoprotein, or LDL, which is low density lipoprotein. So you also get, in this postprandial dynamic phase, remodeling of these lipoproteins into a state which makes them more atherogenic. And we often refer to something called the atherogenic lipoprotein phenotype, which I know is a little bit of a mouthful. But what happens if you have sustained and elevated postprandial lipemic responses, which is what encompasses this change in triglycerides in the blood, you have this generation of these atherogenic lipoproteins. So you get very small, dense LDL, small HDL, for example. And common across both glycemia and lipemia in the postprandial phase is activation of inflammation. And that's what we've been studying as well as part of the Predict program of research. And I think, in answer to your question around kind of the acute and the long-term inflammation and health outcomes, I think it's really important to say to all the listeners that inflammation is a normal physiological response. It's our body's natural defense to an attack. So inflammation occurs after consuming food. It occurs after exercise. And when it's local as well, which is kind of an acute inflammatory response, let's say you cut your finger, you bleed, you have slight swelling, that's your local inflammatory response. It resolves very quickly and it's a favorable response. The problem is it's a double-edged sword. So in the wrong place, at the wrong time, and in excess, it has undesirable consequences. So I think we need to hold that in our mind that yes, inflammation can be good, but it needs to be in the right place at the right time and for the right duration and the right size. Once we start to deviate from this, this is where you see it become this kind of common feature of many different chronic diseases, so type 2 diabetes, cardiovascular disease, and some cancers. And so what we know from our own research is if you have more of this postprandial, so post-eating food inflammation, you have a higher risk of cardiovascular disease. And we see this from our own cohort as well.
I appreciate the clarification on inflammation, right? This is such a general process, one that is essential. It's not automatically associated with concern, right? So after the Buccaneers won the Super Bowl and we have Tom Brady, right? This elite athlete who's playing well into his 40s. And then in his interviews, he's like, everyone wants to know, like, man, like, how do you do it? Are you drinking like the magic waters, this, that? And he's like, oh, I don't consume any inflammatory foods. And then of course, like following that, there's a whole bunch of questions like, what are inflammatory foods? Like, is inflammation bad all the time ever? So I think differentiating that this is a natural process and there are differences in concern to what's going on acutely and on a chronic basis. It's valuable to clarify those roles. And I also really appreciate how you're putting this emphasis on appreciating the entire postprandial metabolic response. I don't know how active you are on Twitter, but there was like this recent ruckus surrounding whether healthy people should use CGMs. Are you familiar with that?
I have seen it and I am watching with great interest because we're currently working on a paper looking at exactly that with the hope of answering that question. The problem is there just isn't enough data yet in healthy people apart from what we have with the Predictors studies. So we do hope to be able to answer. And I think an important point, and this brings in what you just said about the inflammatory response, is that an increase in circulating glucose, an increase in circulating triglycerides is also a normal physiological response to eating. You don't want to flatline, otherwise you would be dead. It's a normal physiological response. But what we are starting to understand is that even within healthy people, these big excursions that you get in glucose, for example, after having carbohydrate-rich meals may cause an increase in inflammation, may cause an increase in oxidative stress, and may damage the lining of our blood vessels, which is called the endothelium. And there's some evidence emerging around this. And so whilst I'm not in any way ever advocating that someone should fit a CGM and try and flatline, I think it's a case that we need to watch the space. We need to look at the evidence that's going to be coming out over the next coming months from the Predict studies. But I do believe that there is a place for CGMs in healthy people, but we must not become obsessive. And, I'm always telling people that we really have to focus on not just how healthy the food is, but the pleasure of the food. It's a social, it's a cultural, it's an emotional event. And we must enjoy our food. And that's the only downside, I think, of if people become too obsessed from self-monitoring, that they forget that food is there as a pleasure as well.
Absolutely. And that seems to be the central concern around this, right? So basically there's limited evidence available to demonstrate the efficacy. And then also perhaps, there becomes this obsession with keeping glucose fat line. I mean, I've seen some obscure takes on the topic from people in like the ketogenic community who've obviously gotten fascinated with their CGM data, and they'll try to demonize foods like oatmeal, right? And they'll be like, look at this spike in glucose following oatmeal. Like the government lied to you. This is not a healthy carbohydrate. It's completely ridiculous. But then following this obsession with postprandial glucose, there's just this complete abandonment of postprandial lipemia. And it's very interesting because, I mean, this is a robust body of evidence looking at, postprandial triglycerides and the association with cardiovascular disease. And I think that is a valid concern as well, right? Like, people start substituting apples for bacon, thinking that this is the way to go. But why do you think that is? Because I'm sure you've noticed the same thing, right? Where people, they don't talk about postprandial lipemia. They don't
Yeah.
recognize its utility. Is it just because at the moment it's not able to be practically measured, or what do you think?
So I think you've kind of hit the nail on the head, that it's very difficult to measure. So I've spent the last 20 years of my career studying postprandial lipemia. It's blimmin' exhausting for me as a researcher doing it. It's burdensome for participants because of the time frame. With glycemia, you can do finger prick, you can do CGMs, you can cannulate someone, so put in a fixed needle, but you only have to do it for an hour or 2 hours. With lipemia, the only option is to be collecting venous blood over an 8-hour period, because we know that it has this really long time period. Now, we're at a slight transition that there is now the ability to look at postprandial lipemia using dried blood spots, and this is some relatively new technology, and the first time it's been used in a kind of personalized nutrition remote way before with our Predict studies. But it's really burdensome, it's really expensive, and I think this has been a big challenge. I think as well, the science has been a bit more delayed on lipemia compared to glycemia. And because there's been so much emphasis on glycemia because of us trying to disentangle all the different problems that are going on with people with type 2 diabetes. We know, though, that there's some reasonable, robust evidence from some of the really big prospective cohort studies that show that postprandial lipemia is an independent risk factor of cardiovascular disease. We also know from mechanistic studies why. This lipoprotein remodeling that I talked about, the inflammation, for example. So we are making huge leaps, and the Predict study is, I think, a study that's really going to push the boundaries in terms of our understanding of postprandial lipemia, because prior to this, the lipemic studies have typically been, a very small scale.
Absolutely. So that brings us into the first recent publication that I want to discuss with you, and that is the association of postprandial lipemia and postprandial glycemia with this inflammatory response. And before discussing the results, I think it's probably a nice way to start by talking about how you measure this, right? So we had these two different factors in terms of a more traditional marker, which was IL-6, and then a novel one, which was glycoprotein acetyls. I'm not sure if I'm pronouncing the second word correctly, but for the listeners who might not be familiar with these markers, especially the latter, could you maybe touch on why this is something we should be interested in and was a good candidate to be used in measuring this response?
Yeah, so let me first say how we undertook the study, and then I can talk about the measures. So for the specific research on inflammation that we published, we used our clinic day data from our Predict-1 study. So we had participants attend our clinic fasted. We took a fasting blood sample, which we measured these inflammatory measures, as well as fasting glucose and fasting triglyceride levels. We then gave participants a high-fat test meal for breakfast, another and high carb, another high-fat, high-carb test meal 4 hours later for lunch. And over sequential periods of about every 15 to 30 minutes, we measured circulating glucose, circulating triglycerides, and at certain time points, we also measured inflammatory markers. The two measures that we used for inflammation were IL-6, interleukin-6, and the other was glyc-A. Now, IL-6 is quite a traditional inflammatory measure. It's synthesized in tissues, so in the adipose tissue, and also by the endothelium, which is the lining of this blood vessel. So it has a very rapid initiation, very rapid onset, so it increases very rapidly. We chose to measure this one because this is the one measure that many studies have shown changes postprandially after consuming a mixed meal. But there has been some question in the literature as to whether it's released actually due to damage to the blood vessel during the blood collection process, so that there's a low level of fasting because you've only just put the needle in, but it increases at later time points due to damage to the endothelium. So we also wanted to look at a very robust measure that's not actually too prone to changes due to maybe small variables such as that. And there's this measure of glyca that is a novel emerging measure of inflammation that's possible to be detected using NMR spectroscopy. Please don't ask me to go into any detail on the methodology of that. But basically it's detecting a signal from a certain group of proteins that allow us to look at a systemic, so an overall kind of global inflammatory response that's going on in the body. It's a really good measure because it's stable. There's really low variability within individuals. So people often hear about something called CRP, C-reactive protein, when we look at inflammation. That's really variable. I could literally just have a sniffle or maybe a mouth ulcer. My CRP could go sky high. So it's actually quite difficult to use in research unless you've got, beyond thousands of people. So this is one advantage of glyca, is how stable it is. It's also, there's very low variability in the laboratory methods to measure it. So it makes it a very precise measure for us of this kind of global inflammation. And more importantly, we know from some of the epidemiological studies that are coming out over the last few years that it's one of the single most strongest measure of circulating biomarkers that's associated with some chronic diseases, so cancer, fatty liver, cardiovascular disease, and type 2 diabetes. So it works really well for us in that it's a very stable measure. We know that it's a measure of global inflammation, but also we know that it's very strongly associated with disease, which is why we specifically looked at this as well as this traditional measure of IL-6. Now, there's lots of other circulating inflammatory measures we could have looked at as well. As always with all studies, that we have to pick and choose based on budgets, based on burden and how much blood we can collect.
Very good. Now, what did you end up finding in regard to these markers and the response to these standardized test meals?
So we found huge variability. So just in line with all of our other measures, such as triglycerides and glucose, we found that there was a huge variability between individuals in their inflammatory response following these standardized set meals. So remember, this is in a clinic setting where it's really tightly controlled, where people are consuming exactly the same meals, but we still saw about 20-fold difference in people's glyca or IL-6 inflammatory response. We also looked at the relative contribution of the postprandial lipemic, so the kind of blood fat response, versus the postprandial glycemic, so the blood sugar response, in the initiation of this inflammatory response. And this again fits in with the whole premise of what we're trying to do with the PREDICT studies, is always look at the relative contribution and the integrated response. And what we found is that it was the postprandial lipemic response that was a stronger determinant of the postprandial inflammatory response compared to the postprandial glycemic response. Now, both of them were still important, so it's not by any means saying that glycemia had no role to play. So the co-occurrence of high glucose in the blood and high triglycerides in the blood is not going to be great. But what was really interesting is that we found that the postprandial lipemia, so the circulating fat, was actually the strongest determinant. We also found that visceral fat mass, so this is the fat around our tummy, was really strongly associated with this response. We found also that there was a higher response in males, higher response as you aged, and also a higher response in postmenopausal women. And then the last thing that we did as part of this work was try and look at those that are having a higher glyca response, so a higher postprandial inflammatory response. How does this relate to their cardiovascular risk? And what we did was we separated out those people falling in the top 10% of their inflammatory response versus those that had the lowest rise in inflammation. And we found that there was about a 2-fold difference in their predicted cardiovascular risk. So those that had a higher postprandial inflammatory response had about 2 times more likelihood of developing cardiovascular disease compared to those that had the lowest response.
The between individual variability is just so fascinating, right? And I think, as mentioned in probably your article, like there were healthy people on paper, and some of them just couldn't deal with the amount of lipids in the meal and clearing that within a reasonable time frame, whereas others had no issue whatsoever. And you mentioned a couple of factors to consider, but when it comes to the general healthy person and the potential implications of these findings, it is very thought-provoking, right? Like, I mean, what could we practically do with these results? For if you found a healthy individual who had a tough time clearing the lipids from this meal within this, I believe it was like a 6-hour window this was measured. I mean, does that
Yeah.
suggest for that person maybe a relatively lower fat diet might be best suited for them? But again, I'm not necessarily I don't know their postprandial glycemic response. I can't assume that was good while they had difficulty clearing lipids, right?
Yeah, I think there's two tactics we can take with this. So one is you can diminish what's actually causing that inflammation. So one is, how can we attenuate the lipemic response and also the glycemic response? And so thinking about strategies to reduce that. And this isn't about replacing one nutrient with another. And I think it's really important to note that, just, as you alluded to earlier about, the low fat and low carb mantra that goes on between people and certain groups. It's about thinking about how we can manipulate our foods to attenuate this postprandial lipemic and postprandial glycemic response. And a key tip, and something I often talk about, and something that's been an area of my previous research, is all around the food matrix. So we know that the structure of food is really important in determining how we respond to food, and really important in determining our lipemic and glycemic responses. And if we can consume food in its original matrix, then we can really attenuate those responses. And I can give you a couple of quick examples if you'd like. So we've done research at King's together with other collaborators where we fed people whole rolled oats. So they're obviously not in their total original matrix, but it's still not very processed. And then we fed people really finely ground oats, so really processed oats. And what we find is that the glycemic response to the whole rolled oats is significantly lower compared to these highly processed oats. But nutritionally, on the back of pack labeling, they have exactly the same nutrients. All that's changed is the matrix, so the food structure. If we take a fat example, we could take almonds, for example, which are eaten as whole nuts or as almond butter. And we know from research that I've done, if we were to look at the lipemic response, if we feed people whole almonds, so in their original matrix, the original plant structure, versus grinding them all down so that they're processed or the structure's broken, again, you have a significantly lower lipemia from the whole almonds than the ground almonds. But again, on the back of pack labeling, they would have exactly the same nutrients. And so there's lots of different strategies we can put into place. But I think the key things to mention about attenuating your lipemic and glycemic response: consume food in its original matrix where possible, or minimally processed. Consume fiber-rich foods, and consume a whole balance of food. Have balanced meals that contain some fat and some carbohydrate. So that's one strategy. Another strategy is we can actually attenuate downstream the subsequent inflammatory response. So we know that there's lots of nutrients, or what we often call non-nutrient bioactives, such as polyphenols, that are found in foods, that even when we've kind of set that fire off of inflammation, we can dampen that fire. So if we consume, again, a variety of foods that have a very colorful with these polyphenol pigments, that cause this pigmentation in foods, then we can attenuate this downstream fire from this increase in lipemia and glycemia. So that's two strategies. And something else, Mike, I just want to pick up on, when we were talking about the value of CGMs in healthy people. I think this research, and particularly in relation to inflammation, where it's in healthy people, but we're still seeing this huge variability, shows you where there may be some value in healthy people with CGMs, because it's all very well using it for people once they have some sort of disease like type 2 diabetes. But what about those people that we want to kind of get a peek into the future? And I really think that by looking at postprandial responses, it gives us that peek into the future. So we will have many people whose fasting glucose, fasting lipids, fasting inflammation is the same. So they look the same on paper if we were to measure their fasting levels. But if we measure their postprandial levels, of either glucose, lipids, inflammation, oxidative stress, etc., to a standardized meal, that's where you start to see this big discrimination. So it's kind of like this kind of black box that also gives us this peek into what may happen 10 or 20 years later down the line.
Yeah, I think that's extremely important as well. I mean, not everyone was a fan of Peter Tia's article on CGMs for healthy people, but I thought he brought up a few really important points. And basically, you know, the average person is in the postprandial period for a much larger portion of their day than they are the fasting period. And it's really important how they are responding to their meals throughout the day because in the case of markers like HbA1c, right, like that can be fine on paper, but there can still be a lot going on within the day itself that warrants room for improvement. And then even when it comes to triglycerides, right, based on what I've seen in the data when it comes to non-fasting triglycerides, I mean, it's definitely at least as good of a marker as fasting triglycerides, and in many cases is probably preferable and gives us more insight into risk than just these fasting values.
Yeah, and the big epidemiological studies support this, that looking at these postprandial values is more predictive of risk than looking at fasting values alone.
Before we leave the topic of postprandial lipemia, are there any consistent trends in the literature when it comes to the type of fat that's within this quantity? So if we were to look at, 25, 30 grams of saturated fat compared to monounsaturated or polyunsaturated, it seems like there's been conflicting findings, but from someone who actually has been studying this for decades, I would love to hear what you've noticed in this area of research.
Yeah, so one thing we can say for sure is there's a dose response effect. So that, and in my answer to your question, these questions are going to be in the context of at the average, the mean person. So on average, there's a dose response effect where the more fat you consume in the meal, the higher your lipemic response. In terms of the fatty acid composition, so by this, the saturated mono polyunsaturated fatty acid composition, there's no clear consensus. And the consensus has not gained any clarity over the 20 years that I've been studying what determines postprandial lipemia. And that's been very much the focus of my research, is how the different fatty acids impact our lipemic response. What we do know is that omega-3, so they're very long chain omega-3, so DHA and EPA that's found in oily fish, that does attenuate your lipemic response. But what we don't know is whether saturated fatty acids cause a higher lipemia than monounsaturated, for example. There's lots published on this, but every study often contradicts the other study. Now, the reason I believe that this is the case, and this has been the focus of my work over the more recent period, is because it's become evident that the food matrix is what's important here. It's also evident that the melting point of the fat is really important. So if the monounsaturated fatty acid is within a high melting point fat overall, because remember, a fat is always a mixture of different fatty acids. So we might call a fat saturated rich fat, but actually it still has some mono or polyunsaturated fatty acids. So if the monounsaturated fatty acids are within a more solid fat, then we know that if the fat is still quite solid at body temperature, it attenuates the response. And vice versa, if we have some saturated fatty acids that are within a fat mixture that's liquid at body temperature, that's going to cause a higher response. We also know, and the almond example I gave earlier is a perfect example of the matrix that the food is in is actually really important as well. So this is why there hasn't been that clear consistency, because it's difficult to disentangle what's the effect of the fatty acid versus the melting point of the fat mixture versus the actual food matrix that the fat is given in. And this is why for my studies and the Predict studies, we use muffins as a vehicle to deliver the fat, because it's a very processed, highly refined matrix that we know we're not going to have that added complexity of the kind of plant cell structure that you get, for example, with nuts.
Excellent. Dr. Berry, last topic for us today as we're coming up on time. your publication on postprandial glucose dips and how this relates to hunger levels throughout the day and overall energy intake. I saw a lot of conversations about the results, but I want to hear it from you, the people who actually studied this. Run us through the study and what the main findings were.
So this study was using data from our at-home phase of the Predict 1 study, where participants wore continuous glucose monitors. They had standardized meals, as I mentioned before, for breakfast. And then for the rest of the day, they had free living, free choice meals. So they would have meals in the morning that differed in their fat, protein, carbohydrate, and fiber content. Then they were asked not to consume anything for 3 hours. And then after 3 hours, they could then consume whatever they wanted for the rest of the day. And what we monitored was different features of the glycemic response. And I think this is really important to kind of pause on for a moment. Typically in research, we use very standardized features of a response to food. So typically in the past, we've looked at 6-hour area under the curve to look at lipemia. Typically, we look at either the peak glycemic response or 2-hour area under the curve for glycemia. But what we now know is that actually it's really important to pick what feature of the response curve we want to look at in relation to what output we're interested in. And so with this research, what we did was we did look at the traditional features. So we looked at the 2-hour area under the curve, we looked at the peak glycemic response, but we also looked at the dip in glucose. And this is the dip that occurs in quite a large proportion of people, dependent on the meal, 2 to 3 hours after you consume a meal. So if you may remember, I said when you consume carbohydrate in a meal, you have a peak at 30 minutes, it returns to baseline around 2 hours. And quite often you get this dip between 2 to 3 hours in your glycemic response. And so what we did was after these standardized meals, but also all of these free living meals, we looked at what was the most important determinant of factors related to hunger, satiety, energy intake. Was it the peak concentration? Was it the 2-hour end of the curve? And was it the dip? And what we found was that people's dip, so this dip in their glucose below their baseline level at fasting, was actually the most important determinant of, firstly, how hungry they felt. Secondly, their energy intake at their subsequent meal and their energy intake over 24 hours. And also how quickly, so time to the next meal, how quickly they wanted to eat. And then what we did was we then separated people as to whether they were big dippers or small dippers. So we got the whole group, our whole cohort of 1,100 people, and we separated them according to quartiles. So we separated them into 4 groups, with the quartile 1 being the people with the least dip, and quartile 4 being the 25% with the highest dip. And then we compared the highest quarter or quartile of people with the lowest quartile of people in terms of their dip response. And what we saw was that the big dippers, so these 25% in the highest quartile, had about a 10% higher increase in hunger in that postprandial period. We also found that they also ate on average about half an hour before the little dippers. We found that in the next meal that they did eat, they had about 75 more calories, and over a whole day period, they consumed about 310 calories more. And I think that's really important because, you know, typically we do know what you eat in a preceding meal might determine your energy intake in the next meal or your response to that preceding meal. But typically over a 24-hour period, our bodies are really clever at kind of making up the difference and keeping that set point. And I think for me, this was really exciting alongside, you know, these differences in hunger and time to the next meal, is the fact that over the whole 24-hour period, we still saw this big difference of up to 300 calories between these big and little dippers. And if we equate that to what that could mean over a whole year, you're talking about a 10 kilogram difference in weight gain or weight loss between big and little dippers. So yeah, I thought that was really exciting, and also a really nice illustration of how we must be mindful of looking at different features of the glycemic response depending on what we're interested in.
Yeah, I would say that finding on 24-hour energy intake and that significant difference between big dippers and little dippers is really what stood out to me. when we consider these findings in relation to the overall body of evidence, which probably isn't as large as we would like it to be, right? I think this is a topic that even the average person who knows a little something about nutrition is fascinated with, right? Because we always hear about these fluctuations in blood glucose, and maybe you're
Yeah.
getting a little bit hangry, and how does that affect meal frequency and energy intake over the course of the day? And it seems like from the limited evidence available, the findings have been mixed, and not as eye-catching as your own. So when we look at what your trial did compared to other ones, I think there are probably some clear differences that are worth considering in interpreting these results. Like when when I think about the evidence, a paper that comes to mind is Melanson, 1999, and they had like a 7-hour period, and they looked at the response to a carbohydrate drink and a fat drink, and how that affected energy intake at ad libitum meals, and when participants request for food. And you see within there that after the fat drink, the request for a meal was about like 1 to 2 hours after the carbohydrate drink. And even, satiety scores were higher and hunger scores were lower with the fat drink. But then you don't see these differences in energy intake over the feeding window. And then of course we have like these other studies, and they're looking more in an acute sense, right? So they might look at a drink and then maybe like 2 to 3 hours after, as you mentioned, but they're not really looking at the whole day, how the whole day is affected. And I think that's what really stands out about your paper, is that you looked at the 24-hour differences, because over 2 to 3 hours, it was what, like a 75 kcal difference or so. And that doesn't really
Yeah.
stand out. But like 325, that's a serious surplus. So I guess tell the listeners, how you interpret those findings and how they relate to other evidence on the topic, and maybe what we still need to figure out here.
Okay, so I think anecdotally, we're all probably aware that, you get a sugar crash and you feel hungry, or, I know my 8-year-old son is constantly experiencing hunger, and, we know either from ourselves or if you've got kids what it's like. And I think there's lots of anecdotal evidence, but actually when you delve into the literature, there actually isn't that much evidence in human participants, particularly in a free living environment. I know you pointed to one study, and I know that there's mechanistic studies, and we know that, the different drivers of hunger and our gut peptides. I think what's different with the work that we've done, and I think builds on what has been done before, is that firstly, we're looking in a free living environment. We're not looking in this very tightly controlled clinic setting. Secondly, we fed standardized meals that were isocaloric, so they had the same energy content. So you referred to a study where you gave either a high fat or high carb. So it's difficult to disentangle here whether it's just the nutrient composition of the meal that's causing these differences, or whether it's actually the dip. And so what we were able to do is we were able to look within standardized meals in duplicate, so where people had exactly the same test meal, so the same carbohydrate or same fiber, as well as look with these ad libitum free living meals. And what we could see by looking within these standardized meals is even when you're feeding exactly the same nutrient composition, you still get this difference, because there's huge variability in who's a different, who's not. Once you separate them out, you get this difference in, in this hunger and this energy intake. So we can see that it's due to the dip, not necessarily just due to the composition of the meal itself. And what we could also do is, because we were able to, as I said, give these meals in duplicate, we were able to tease apart person factors, environmental factors from the meal factors and the dip factors. And I think this is what this really, builds on this, to show that there does seem to be this independent effect of the dips. And I think that also having the free living, like I said, is really building on what's been done before. So the work that's been done before has, really plowed the way to enable us to actually, design the study that we did and make these interpretations from our data.
Fantastic. Dr. Berry, that is all I have for you today. This has been a wonderful discussion. I really appreciate your time. For the listeners who want to go and learn more about you and support your work, where can we point them towards?
So you can follow me on Twitter at Sarah E. Berry, also on my King's website. And also if you want to find out more about the Zoe studies, there's the joinzoe.com website that has all the details up to date on all of the Predict publications and the Predict program of research.
I'll link those resources in the show notes. Highly encourage everyone to check out this fascinating work that is going on. That does it for another episode of the Muscle Memoirs podcast. Thank you so much for listening, everyone.